Whether you're domesticating Golden Gate parts, building a library, or modifying a protein, site-directed mutagenesis, or SDM, offers the control required for modern molecular cloning. Today we're breaking down the different types of SDM, including multi-site protocols, that you can run cheaply on your plasmid. We'll be using PCR, and I really don't want to bore you with that. Let's jump right into the SDM techniques. Substitution. This is your go-to method for domesticating Golden Gate parts or building a library. The primers are designed back-to-back, with one containing a mismatch that gets integrated into the new molecule. The substitution can be placed almost anywhere in the oligo sequence, but it is best practice to keep it in the middle to provide a more stable flanking region. The melting temperature is based on the regions that match the template. Next is deletion. This is the easiest way to remove an unwanted sequence. Here the primers are designed flanking the target region. The polymerase simply skips that part, and once the product is circularized, that segment is gone. Finally, insertion. This lets you add new sequences. For small additions up to 5 nucleotides, attach the new sequence as a 5' overhang on one primer. For inserts over 6 nucleotides, split the sequence between both primers as overhangs. As with substitutions, calculate your TM based only on the template matching regions. To explore multiple variations at once, you can build a library using degenerate oligos at your target site. This is a massive time saver, as it avoids the need to design and order thousands of individual primers. With just one PCR reaction, you create a pool of variants ready for screening or directed evolution. Just remember, the same rules from standard substitutions apply. Regardless of which mutation type you choose, the actual PCR setup is the usual. This is one example. I personally prefer using a commercial 2x high-fidelity master mix, especially hot start versions. However, any high-fidelity polymerase will work. Once your reaction is ready, you use the standard PCR protocol. Pay attention to the annealing temperature and extension time recommended by your kit. After PCR, you need to process your linear DNA before transformation. Use a mix of T4 polynucleotide kinase to phosphorylate the ends. DPN1 to remove the methylated template, and ligase to circularize the plasmid. Using a commercial master mix for this step can save you time. However, if you opt for the homebrew method, simply add 0.5 microliters of each enzyme, and incubate for 10 to 15 minutes, at 37 degrees Celsius to get your product ready for transformation. If you need a more versatile or budget-friendly route, consider the Liu method. This technique maintains the simplicity of a single PCR reaction while offering incredible flexibility. It isn't just for point mutations. It can handle insertions up to 100 BP and deletions in one go. Perform multi-site mutagenesis, and it is incredibly fast and cost-effective because it requires fewer PCR cycles, and only DPN1 digestion is required after the PCR is complete. The efficiency of the Liu method relies on the specific primer architecture. A 5' overlapping region, of 10 to 15 nucleotides, containing the mutation, and a non-overlapping 3' region, with a TM of 5 to 10 degrees higher than the overlap. This drives the initial amplification, preventing off-target primer-dimer formation. The high TM 3' anchors ensure the polymerase is already moving before the 5' overlaps have a chance to interfere with each other. This design generates fragments that anneal during cycling to form nicked circular plasmids, effectively mirroring circular polymerase extension cloning. This mechanism is particularly advantageous for multi-site SDM, as multiple overlapping fragments assemble into a single plasmid in a one-step reaction. Both single and multi-site mutations are performed in a 50 microliter reaction volume. While the template concentration remains relatively consistent, for single-site mutations, a 500 nanomolar of each primer is used. However, in the multi-site protocol, the primer concentration is reduced to 100 to 200 nanomolar to minimize the formation of primer dimers. The extension time for single-site mutations is based on the entire plasmid length, while for multi-site mutations, it is based on the longest fragment. Where the PCR changes compared to back-to-back oligos is the number of cycles, which is 15 to 18. This lower cycle count is sufficient due to the high efficiency of the assembly and helps minimize unwanted secondary mutations during the process. After PCR, The only step remaining is the DPN1 digestion, which you add directly to the PCR product. There is no need to phosphorylate the fragments or perform a ligation. The product is already in a circular form, and E. coli will repair the nicks following transformation. Mastering these techniques gives you total control over your genetic constructs. If you found this breakdown helpful, consider subscribing. My cat says happy cloning. And I'll see you in the lab.